You are listening to the Plant and Animal Cells Virtual Lab instructions. In order to complete this assignment, you will need to have the lab handout, which looks very similar. Make sure that you have the lab handout before you begin. The other thing you're going to want to do is pull up a copy of these instructions on Google Classroom because you'll need access to some of the links. You might pause the video to take a moment and open up that document now. The guiding question for today's virtual lab is, how are plant and animal cells alike, and how are they different? Procedure Part 1 is animal cells. Access the virtual microscope at this link. You're going to need to go back to your classroom document and click on this link to open up the virtual microscope. Once you click on the link, it will bring you to this page, and you'll need to click right here where it says the virtual scope. You may need help unlocking your Adobe Flash Player. If so, just raise your hand and I will come around and give you some help. So you're welcome to take this tour and let the program explain to you how to use the microscope, or if you'd like, you can just follow along with me. But this is basically like an actual microscope, and it's going to show you the different types of adjustments that you can make on your microscope to help you view things through the lens. We're going to go back to the instructions here. It says, view the slide labeled cheek smear. Draw the image at scanning, low, and high power in the boxes below. So on your handout, you've got 3 different boxes with circles in them. This is representing what you're going to see when you look through the microscope when you're looking at it zoomed in by 4 times. So this is a power of 4. This one is the power of 10, and this one is the power of 40. So it's like you're zooming in closer, closer, and the closest you'll be able to get. Once you sketch what it looks like at 40 times its original size, you're going to label the cell membrane, the cytoplasm, and the nucleus. You really won't be able to see those at these lower powers. So let's take a look at how we can do that on the microscope. So here's your microscope, and it says the cheek smear. So if you look at these are all different slides. You've got the letter E, onion root, bacterial capsule, and a cheek smear. So this is the one that we want to add to our microscope. So if you click on it, it'll automatically put it on the microscope for you. Now, over here, if you get lost and you don't know what to do next, there is a checklist that you can click on, and notice that it gives you all the things you need to do. So right now we've chosen a slide and we've got it at the 4 times lens, which is perfect because that's what we want to start. So now we're going to need to turn the light on. In order to turn the light on, you're going to need to scroll down. The button is right here. And notice if I turn it off, so this is where the light's going to shine through. The light comes from the bottom of the microscope and shines through the sample. So notice that light comes on. All right, so now we've got our light on. Let's take a look at our checklist. The rheostat needs to be at 10. So we're going to scroll down, and right down here, we can just click and drag this until it says 10, and that's going to change the rheostat for us. All right. The specimen, that means this little sample, needs to be centered. And we're going to open the iris temporarily in order to do this. So the iris is this little guy right here. So we can open it all the way to let as much light in as we possibly can. And then we're going to adjust where this sample is. So this little blue sample needs to be right over the light source. So we're going to use these different rollers to help us get it exactly where we want it to be. Oops. There we go. So you're going to use these top 2 rollers. And now you can see that little blue dot is kind of right above the light source. Then we can close the iris again. All right. Now we need to start the stage at the top position. So this part that has the sample on it is called the stage. We're going to move that all the way up. Okay, so notice how you can move it down and then back up. So we're going to start it all the way at the top. All right, the next part of our checklist is to adjust the oculars. The oculars are basically these 2 little eyepieces. So we're going to adjust those. Adjust oculars only while looking through the scope. Turn light on and switch views. All right, so in order to adjust these eyepieces, we're going to need to switch views. So I'm going to close this for a moment, and right here it says switch views. So now this is like looking through the microscope. So it says you must adjust your oculars before viewing the image. So we're just going to click and drag these, and notice at some point you get a view where it's just one circle. So you want to adjust those until you just have one circle. That looks good. And we're going to switch views again and look at our checklist. Now we need to adjust the coarse focus. There are two focuses. One is coarse and one is fine. In order to do that, we're going to switch views again. And this one is our coarse focus. And as you click, so you just try a couple different ways. Try going up, try going down, until you have some kind of image. Notice that the image disappears when you get too far. So this would be the rough view. Okay, notice it's not very well focused. So next we're going to have to use the fine focus to adjust that. So again, you can just go up and down, just click this little roller. That seems to be getting fuzzier, so we're going to go the opposite direction. You want to click until you get the most clear view possible. Now, I will give you a little hint. When this red circle shows up, you know that you've done it correctly.
Rain Alcantar, please come to the office. Rain Alcantar, please come to the office.
Sorry about that announcement. All right, so the next thing you're going to do, you can adjust this a little better so that this red circle is right in the center of your screen. Okay, and you can put it down here on the measuring tape, basically, if you'd like to measure something. Otherwise, you can click it above so you can see it. So basically, this is as good as it's going to get. So remember that you are on the 4x, so 4 times the size view. Now that you've done that, you're going to need to go back to your worksheet, and you're going to draw what you see. So you're going to draw what you see in the little red circle. You're going to take this and sketch it right here. So pause the video, take a moment to sketch that, and then we're going to come back and try the low power of 10. All right, now in order to go back to change this to the power of 10, you can actually do it right here if you like. And you might need to scroll down to see. So before we were on the 4, okay? And so the one that's in the back is the one that we're using. So if we want to roll it, so now the 10 is in the back, notice what that does to our view. It zooms in even further. Now all we really have to adjust is where this circle is. So let's adjust this so it's a little clearer. And then you can use the fine focus to get it even more focused for you. That looks pretty good. So this is what it looks on the 10 power. So now you're going to go back to your worksheet and draw that sketch right here. You might pause the video and come back to do the next power, which is 40. So now to do the 40, we're going to rotate this again until that 40 is right in the back. Okay, and notice we're zoomed so far in that you can barely see the lines of that red circle. So you can kind of adjust this to wherever you think makes a good picture of a cell. So you can kind of move it around a bit until you are satisfied. I mean, this one looks pretty good. Notice this really dark spot in the middle. You can kind of see the outline of the cells. This looks good to me. I'm going to adjust my fine focus just to see if I can get it a little more focused. And again, you're just going to click in one direction or the other until it's as focused as you think you can get it. And then you're going to go back to your worksheet again, and you're going to sketch that one right here. Now, once you're done sketching, make sure that on this picture you label the cell membrane, cytoplasm, and the nucleus. All right, that completes part 1. That's the animal cell. You're going to click on a new video to look at how to do part 2, the plant cell.